ou ceux de l'autre, de nouvelles stratégies dans une réflexion vaccinale. Voilà. Thank you.
Hi. My name is Jim Samuel. It does have good effect on here. So I'm from Texas A&M University. At this institution, the students are called Aggies. And the common response by Aggies, learned response, is howdy. So if I say howdy, you're supposed to say back, howdy. There you go. You're an Aggie now. So I want to thank the people that Dr. Med, who invited me, and the other people in the laboratory for asking me to come to join you today. It's a very kind invitation to come from the U.S. and tell you about what I work on. This is not Texas. It's actually Maui. I just came back from Maui. I've had a delightful spring. And it's kind of like the weather here, though. So you're not much different than Maui. I think you're closer to Maui than Texas is. So what I've titled the talk today is Coxiella Burnetti at a Tipping Point. So there's a bestseller, New York Times bestseller, maybe not Paris bestseller, New York Times bestseller book that was out a few years ago, and it suggested that for dramatic changes to happen, that there has to be an accumulation of important new evidence in that event to cause a tipping point response, and after the tipping point, then tremendous change occurs. And so I think this is where we are in the process of understanding the disease entity that causes Q fever, called Coxiella Burnetti. And the subtitle is, it's a stealth pathogen as we model it from our laboratory with novel strategies to manipulate the host revealed by in vivo, in vitro, and comparative genomics. And one of the ways I chose what I was going to talk to you about today is because of the seminal work that's gone on in this institution for many, many years, including from the labs of Dr. Mej and Jigo and Kepo, as well as Didier Raoult. And they have really set the framework for much of what we understand about host interaction with this pathogen, particularly with the opportunity to look at clinical infection, both as chronic and acute diseases. So what I want to talk to you about is briefly review what I think is a paradigm shift in our ability to approach the organism. And so this is a bacterial pathogen, and the way that people traditionally understand bacterial pathogens is to look at mutants that no longer cause the phenotype of interest, which is pathogenesis, and then track back to identify the gene that's required for that pathogenic phenotype. And that's a tool that we have not had available to us until, well, I think you're allowed to be late, Dr. Wells. And that's the tool that we haven't had available to us. And then I would like to take this opportunity, again because of the unique situation, to talk about a little bit of the stealthy remodeling that we choose to think of when we think from my laboratory about how Coxiella pathogen interaction works. And I want to talk about three topics very briefly. And the reason I've chosen these three topics is because I also had the pleasure to serve as a co-editor with Dr. Mej and two other scientists on a new book that is just published that had some review articles from various investigators around the world. And so I wanted to take this opportunity to provide some data that I think helps us understand our own modeling. And then I want to finish by talking about a topic that I originally identified, proposed in graduate school, didn't have the experimental tools to address it. Dr. Raoult and others then took that idea and tried to show the falsehood or the fallacy within my original proposal, which I significantly appreciated, and then how I reframed that question to try to look from a bacterial pathogen point of view how there might be pathotype diversity. And so, for those in the audience who don't know this organism, Coxiella Burnetti is an obligate intercellular bacteria. It replicates in a terminal phagolysosome-ish, and Dr. Gigot and others have suggestions that maybe it doesn't always replicate in the final terminal phagolysosome, but it's an obligate intracellular bacteria. It's Rickettsia, traditionally associated with other Rickettsia organisms, but in point of fact, phylogenetically, it's most closely allied with the Legionella lopha as a pathogen. It has two phases in its growth in terms of its LPS expression, and we won't talk about that other than to say that we use both phase 1 and phase 2 organisms, LPS rough and LPS smooth guys, to do experiments. It's metabolically active outside of eukaryotic cells, and that's been taken advantage of very recently to make this big transition in the way that we can ask questions. It has a developmental life cycle while it replicates in that cell I won't talk about today. I will talk about the differences in disease from the bacterial point of view, and the majority of the information we know about chronic disease comes from work of Dr. Raoul and his collaborators in terms of understanding that impact of chronic disease. And I'll finish this by saying that from a microbiology, bacterial genetics point of view, the elucidation of the genome was powerful in that we understood that it has a coding capacity of about 2,000 open reading frames, and it has a large skew of its proteins with a very high PI, and at the time we proposed, and didn't really have a way to test, that it has evolved these high PI proteins to help buffer the microenvironment that it lives in the terminal phagolysosome, which is a low pH environment. This points out the fact that while Q fever is underappreciated in terms of the burden to the public health communities worldwide, occasionally there are explosions of Q fever cases, and there was an explosion of Q fever cases in the Netherlands from 2007, 8, and 9. That explosion of cases has now been generally mitigated by investigators and public health support in the Netherlands, as well as with people throughout the EU, including Dr. Raoul's group. But it was initiated, it turns out, from epidemiologic studies into agricultural areas that were intensive in their farming of goats. And those goat farms were right next to urban centers, small urban centers, and it appears as though the burden of Q fever infection, which is zoonotic in its distribution and found in a whole range of domestic and wild animals, led to the environmental contamination downstream from those farms. And so my laboratory has been particularly interested in some very broad questions, and they include these questions: what adaptations has C. Burnetti evolved to allow replication in this parasitophorous vacuole? What different pathogenic strategies lead to the differences between acute and disease in humans? And we think there's a microbiological component to that. And how is C. Burnetti replication in infected cells contained both from the innate responses as well as from the acquired immunity? And we have an interest from the acquired immune side in developing a novel strategy for vaccine development using subunits. Our general model is this organism is a self-pathogen, and it attempts to evade the principal innate killing mechanisms. And as I mentioned before, we've developed a number of tools to address these questions, but the most recent change in ability to grow this organism extracellularly, and with that, then isolate mutants using some genetic tools, has allowed this field to sit on the edge of expansion. So before I give some additional background information, I want to highlight this observation that I made as a graduate student. And so for those of you who are graduate students now, this is a documentation that you can ask a question and still ask the question, continue to try to answer that question 25 years later, which is what I'm doing now. So what I've gotten in graduate school was a collection of cybernetic isolates. There weren't a lot of cybernetic isolates available. And by some primitive phylogenetic tools, I was able to establish that the isolates that came from acute disease cases that we had access to were distinct from those isolates that came from chronic disease cases. And that led to the, at that time, untestable hypothesis that there might be a pathotype variation between those isolates that led to the different kinds of disease. And in subsequent years, Dr. Rule's group and others were able to identify that a number of isolates can actually cause chronic disease, including those that were phylogenetically identical to those that cause acute disease. The alternative doesn't seem to be true, to my knowledge at the moment, is that those isolates that led to chronic disease haven't caused acute disease in humans, and I'll show you data to suggest that they also don't cause acute disease in animal models of human disease. To put some general boundaries around how this organism replicates intracellularly, there was a very nice model provided by Dan Goff and Bob Heinzen in 2007, and I'm not going to provide much data, any data really, to change my opinion about this being pretty correct. And the organism is internalized and then quickly goes into an early to late endocytic compartment, interacts with autophagosomes, and generally replicates in a compartment that has terminal lysosomes, making a terminal phagolysosome component with that. What I will spend some time on is then talking about some interactions with the host, particularly those that we think have significant impact on controlling replication. And so this is a schematic to simplistically represent a eukaryotic cell in this large parasitophorous vacuole that can contain cybernetic and the particular dynamics in that compartment. And so in a general sense, I would say that infection does not immediately activate monocyte macrophage populations that they infect. And I won't talk about a lot of the dynamics because Dr. Mej's lab and others have spent an enormous amount of effort and generated tremendous data sets on looking at specific cell populations and the skew between a general M1 and an M2 response after infection by different populations. But if you treat those infected cells with interferon gamma or TNF alpha, it will drive those infected cells to activate and control the replication. So without that activation signal, they're able to, in most cells, replicate very efficiently, and with that, there's a control system. And we think that the primary controls of this are actually the production of reactive oxygen and reactive nitrogen intermediates in that control. And this is a stylized vacuole that has some of the properties of this interaction. This organism is an acidophile. It only becomes metabolically active once it gets to a compartment that is in the pH range of around 4.5 to 5.5, and it certainly replicates with developmental life cycle variants. And most of the studies support the fact that in this compartment, proteases and hydrolytic enzymes as well as defensive peptides are fully active in that compartment, so it's able to evade those. We don't think that it evades oxygen radicals and nitrogen radicals during the replication. We don't think that its replication is controlled through restriction of iron or through restriction of other nutrients like tryptophan. And So our experiments suggest that tryptophan and iron are not elements that allow it to control its replication. Examples of how this organism is able to avoid the host response includes infection of dendritic cells by virulent phase 1 does not result in DC maturation. I sat down with some students this morning and they provided a lot of data to expand that general phenomenon. So I guess what I would say is that it activates at an intermediate level the maturation of DCs from what I think everybody was saying. I'll show you briefly some data to support that in terms of activation mechanisms. We don't think that TOL4 activation is a mechanism of activating macrophages because we think that TOL4, the lipid A moiety, the cognate ligand of TOL4 is actually an antagonist. And I'll also show you one data set that provides that looks at the balance between anti and pro-apoptotic signals during the infection process. And so the data that we support most strongly that suggests that C. Burnetti, among other mechanisms, has evolved one of its HAMP molecules, the lipid A molecule component of TOL4, is shown here structurally by Rodolfo Toman, which suggests that it's a tetraacylated lipid A molecule, which fits in to the understanding with a variety of gram-negative bacterial lipid A's that act as antagonists. And when we purified lipid A from the entire LPS molecules, we were able to see that it does not drive TOL4 signaling at all. And in point of fact, in a TOL4 reporter cell line, it inhibits the TOL4 activation by another cognate ligand of TOL4, which is E. coli LPS. In the context of apoptotic signaling, I would highlight 3 observations. The first was made by Della Casa Grande. He was in the laboratories of Dr. Raoul Amage. I don't know where he is now. Is he still in France?
In France, yeah.
In France? Yes. And he made the initial observation. One of the early cells, monocyte macrophage populations that we used as the cell line to look at the interaction with host cell is THP1 human cells. And they were able to show that in apoptotic cell death that was mediated in part by the elicitation and the recognition by TNF by the infected cells to drive apoptotic death. In contrast to that observation was a set of observations, particularly by Dan Voth and Bob Heinzen, that indicated that infection inhibits the exogenous stimulation, the extrinsic stimulation of apoptosis of THP1 macrophages, those that have been activated by PMA, and in a primary alveolar macrophage cell population. And they were able to inhibit apoptosis, making the model whereby C. Burnetti inhibits apoptotic signals such long enough to allow the cell to survive and then to complete the large replication cycle. And I'll mention a publication that we just had in PLOS One this last month, that infection in a THP1 monocyte, much earlier in its origin, and in human monocyte-derived macrophages, results in a caspase independent apoptosis early in infection through both the TNF-alpha and non-TNF-alpha mediated process, and it does not inhibit extrinsic stimulation of apoptosis. I think this just points out the balance between pro and anti-apoptotic factors depending upon the individual cells. THP1 cells that have been treated with PMA are really at the terminal end of their activation status, and they can't really upregulate a lot of signals anyhow at that point. Another way that many bacteria, upon interferon gamma activation, are able to control replication is by sequestering of iron. And there was an observation early on by Dale Howe, who has been in Bob Heinzen's lab for the last 10 or so, that C. burnetti replication might be inhibited if you restricted the access to iron. And so that might be one of the societal mechanisms induced by TNF-alpha interferon gamma. And so we took that observation to try to establish how C. burnetti acquired iron and whether that was in fact true. And in point of fact, they actually do have the regulon system that most gram-negative bacteria use to regulate the expression of genes that are involved in iron acquisition. And this just shows a schematic of how gram-negative bacteria traditionally regulate the expression of genes that are involved in acquiring iron, and that's through an iron-binding protein, transcriptional repressor protein, FIR, that binds the FIR box in the operons of those genes. And so we characterized the functionality of a FIR gene from C. burnetti and found that it to be A, not very effective as a repressor, but B, we looked through the genome, found promoters that had FIR box-like sequences and could be regulated by FIR, and then evaluated about 25 different promoters. And it turns out that C. burnetti really only uses that FIR regulator to regulate maybe 5 genes. And it turns out that it doesn't appear that there are many iron acquisition systems that are operational in this organism. Probably, as you'll see in the next slide, because we don't believe that it actually requires significant amounts of iron to replicate. And so we kind of repeated the experiment that Dale Howe had published that suggested that if you sequester iron from an infected macrophage, that you would inhibit its replication. In fact, you do this with inhibitor of iron, and it actually these actually allow the faster replication of C. burnetti, completely opposite from what would happen in most gram-negative bacteria. This Legionella pneumophila is the control here, so if you restrict access to iron with an iron chelator, it slows the replication rate of Legionella pneumophila. This is in a macrophage, mouse macrophage cell line called J774, whereas with C. burnetti replication, it allows them to replicate faster. And there are sub-lines of this J774 which are either spontaneously upregulated in their ability to produce NADPH oxidase, which is the principal generator of oxygen radicals in cells. And in those cell lines, they're actually not able to grow very readily, but if you restrict iron, they're able to grow better in those cell lines. And there's also a D9 line of this J774, which is actually has no NADPH oxidase, and they grow even better in those cells that can't even make NADPH oxidase. And so our hypothesis from that set of data was that iron depletion actually lowers oxidative stress of these bacteria through the interaction of iron called the Fenton reaction to enhance replication. And we tested this actually in an infection model in a mouse to see if this happens in an animal model. And so in that mouse model, we created both a hypopuremic challenge. These were animals that had iron deprived from them, versus a hyperpuremic challenge model where animals had additional iron added to their medium. And in fact, as a measure of disease, those that had hypopuremic developed greater splenomegaly bacterial numbers over time. And in the hyperpuremic model, you had a statistically significant lower replication of the bacteria. This is completely opposite from all other gram-negative bacterial infection models that have done this kind of study. So we think that reactive oxygen and reactive nitrogen are major mediators of the killing that's mediated when you activate an infected macrophage with a cytokine like TNF-alpha or interferon gamma. And we did studies quite a while ago that suggested that in those clone macrophage populations, that if you knocked out the ability to express NADPH oxidase, you increase the replication rate, And if you inhibited in these infected cells, if you inhibited either the reactive oxygen or reactive nitrogen burst with pharmacological inhibitors, you could increase the replication rate dramatically. We also did infection in knockout mice and showed that both FOX knockout and reactive nitrogen INOS knockout mice had a better replication and disease process than wild-type mice, and that the double knockout actually had a much larger infection burden. There's negative data to still question this observation, and that is that chronic granulomatous disease mice, or monocytes from patients, were originally found no difference in their ability to support replication. That was a study done 12 years ago, I believe, or longer ago by Maniac Pariah. I think that we'd have to rethink that experiment to test that. But what also supports our general observation that oxidative stress is critical to cybernetic replication is the ability to grow this in extracellular media. And the extracellular media, called ACCM media, published about a year and a half by people in Bob Heinz's lab as well as my lab, were able to show that cybernetic is actually a microaerophile in its evolution. Additionally, because we think that oxidative stress is important, cybernetic appears to have evolved, like a number of other gram-negative bacteria, an acid phosphatase that can inhibit the release of preformed NADPH oxidase that generates reactive oxygen in PMNs from humans. And so this experiment, which was originally from data that Oz Bacchus showed, and then we confirmed his observation, is cybernetic infection doesn't stimulate ROI release by PMNs, but it can inhibit PMA-induced ROI release by PMNs. And so we eventually identified a gene that encodes acid phosphatase. We characterize it enzymatically, and then we're able to show that the recombinant acid phosphatase is able to mediate this PMN inhibition. And we believe that we were able to create a model whereby we track the targets in terms of the acid phosphatase activity, both from a PMA generation and an F-method-V generated signaling system, that these acid phosphatases prevent the assembly of the components at the membrane of the NADPH complex. So then you can ask the question, well, is cybernetic, totally defective in the ability to detoxify reactive oxygen? And so, if you remember from the work I told you I did in graduate school, which was look at these different strains, and I'll follow up with these groupings later, is what we had identified in general was there were phylogenetic groups of bacteria that came from acute disease cases, and then there's phylogenetic groups that come from chronic disease cases, so I'll generally refer to them as these acute isolate groups and chronic isolate groups. And we did a genome sequencing project to identify the difference genetic capacity between those organisms. And what you see, among other things, is that the gene for catalase in the acute isolates is actually a defective gene. It's a pseudogene. It doesn't code for any functional enzyme. And additionally, in at least one of the isolates that does code for a functional catalase gene actually doesn't code for another important detoxifying enzyme, SOT-C. And so we brought out the genes and expressed the proteins and showed that these genes were in fact functional genes for both catalase, superoxide dismutase, and alkyl hydroperoxidase, which substitutes for catalase in some of those strains. But the observation I'm going to point out to you is that we took the catalase gene that was functional from the Dugway isolate and then put it into the acute prototype, the 9-mile isolate, through two different mechanisms. One mechanism is by delivering it on a transposon, a TN7 transposon, opted into the chromosome, and the other is through a plasmid that we're able to transform in. And what we found is that the complemented CAT-E at the low copy was able to rescue some oxygen sensitivity in this ACCM media shown here compared to wild type. But when it's delivered at a high copy number, it actually is worse. And when you look at an infection model, for example, these J774 cells, that the 9-mile strain that got the low copy of the catalase, functional catalase gene, were better in terms of responding to oxidative stress in those infection models. And in an animal model, you can actually see the bacterial burden increase, hence an enhancement of virulence if you return this catalase gene to 9-mile strain. And so the high virulence strain, 9-mile acute prototype strain, can have its virulence increased by that. And our hypothesis on these data is that a higher expression of this catalase elevates its exposure to oxygen because a product of catalase is the expression of oxygen. And that provides additional products for oxygen radical formation. And we think that this organism is at an early stage of becoming a microaerophile. And so they have these kind of conflicting evolutionary pressures of whether to maintain or get rid of some of these detoxifying enzymes. So with that, I want to go into the second half of my presentation, and that is about comparative genomics and whether there is a pathotype variation. And to do that, I want to first talk to you briefly about some animal models for in vivo studies. And so we've used a couple different animal models to look at virulence, and the first is a guinea pig model. Guinea pigs are really sensitive to infection. Just a couple of organisms delivered through an aerosol route can generate a really nice pneumonia pathology at 7 days. It eventually begins to resolve by 28 days. And if you vaccinate the animal with whole cell vaccine, which is protected, you completely mitigate that disease process. And in those, using that experimental model, I went back to ask the question that I couldn't ask in graduate school, which is there pathotype differences between these strains? And so we chose 2 or 3 different isolates from each of the phylogenetic groups, and then tested their ability to cause fever and death at a higher dose. And what we found is that the acute prototype group uniformly caused fever at a higher dose, caused death. Isolates from one of the 2 genomic groups that came from chronic disease patients caused no fever even at the highest challenge dose, but colonized even at the lowest dose. And then the second group had an intermediate phenotype in terms of ability to cause fever, but never caused death in that animal model. We then used 2 different animal models in terms of challenge, one through an IP route, not physiologically relevant, but certainly is a great colonization model, such that the development of splenomegaly is dose dependent. The higher the dose, the larger the spleens. And so this is an unchallenged animal. This is an animal challenged with a high dose, and this is an animal that's been vaccinated. So it's a good readout in terms of ability to grow. And secondly, we've just developed this aerosol challenge model using a Madison chamber to deliver in droplet nuclei to the lungs of mice. And what we found is that, shown both here and here, is that when you deliver the acute prototype isolates by aerosol route, the animals are less well, less thrifty, lose weight the first week, shown here in these different separate experiments, but they recover. Their pathology at different time points is much more severe than the pathology, and there's no weight loss when you're challenging with isolates from chronic disease. And so we think there's a fundamental difference in the response to chronic infection. And in point of fact, we did an analysis of using bioplexing to look at 20 different cytokine chemokines on these animals that were IP infected in a mouse with different isolate groups. And so the red indicates the response to different isolates that are all from that first genomic group that came from acute disease patients versus chronic isolate groups. And so a number of the inflammatory mediators over time were much more significant in the acute isolate prototype groups than they were from the chronic isolates. And additionally, if you look at this as a snapshot of 28 days after infection, we don't believe that the difference in colonization in the spleen numbers account for the differential infection and inflammatory response. And finally, pictures are always worth a lot of words. And so this shows the difference between challenge with isolates from the acute prototype and a chronic prototype. And this is a SCID mouse, no functional B and T populations. We think of it as an innate responding response. And if you look at the outcome for these animals, they have a sad, pretty prompt death involving cachexia, versus these guys who actually don't lose weight, they get fatter than their litter mates. Eventually they succumb to infection, and if you look at them pathologically at these late, when they're eventually passed, their organs are filled with bacteria. So it's just they don't respond with a strong inflammatory response, and we developed a whole series of readouts that we think might help to explain differences in the response in this mouse model between the isolates that develop an inflammatory response and those that don't develop a significant inflammatory response. So, I mentioned before that the differences between these strains led us to engage in genome sequencing event experiments that look at the distribution of genes and the plasticity of the genome between these different isolate groups. And among the most significant observations we made in the differences between these has to do with we think that the major differences have to do with phenotypic variations due to secreted effectors using two secretion systems. And I'll point out one briefly, and then I'll finish off by telling you our observations with the second secretion system. So there is something called a type 2 secretion system, and those are require a sec-dependent secretion across the cytoplasmic membrane, and then use a pilus structure to do a secretion across the outer membrane. And there's one called type 4 pilus structure, type 2 secretion system, that's well characterized in Francisella. It's kind of unique among the type 2 secretion systems. It turns out that there are pseudogenes predicted for some of the important components of those in the isolates that come from chronic disease patients. And we have secretion studies to support that there's a fundamental difference in their ability to secrete, and if you complement the defect with a wild-type gene from 9 mile, then you can complement the secretion phenotype. And I'll point out that in the Legionella and Haemophila, in the Francisella and Legionella and Haemophila field, the type 2 secretion elements are able to secrete a number of critical enzymes through their system. But the most important secretion system evolutionarily, I think, for this bacteria and many intracellular bacteria are these type 4 secretion elements that are able to secrete effector molecules into the cytoplasm and infected cells. And we benefit from our near neighbor, Legionella and Haemophila, in that 23 out of the 26 components of the .ICM system in Legionella and Haemophila are homologous genes in C. Burnetti, and significant number of them are able to complement by the smiley faces, and a significant number of them aren't able to complement the defect in Legionella when they come from C. Burnetti versus the unsmiling faces. But this similarity in readout provided us with an opportunity, us and others, with an opportunity to then look to see if we could identify effector molecules that were released. And so there was 2 important observations that were published this summer. One from Craig Roy's lab and one from Bob Heinzen's lab, and both of them identified that mutations in one of these key components of the .ICM system made those organisms unable to replicate intracellularly, but were able to still replicate like wild type in this extracellular media. And we made similar observations using both the dominant negative that we constructed in Coxiella Burnetti, as well as we have a number of transposon mutants that also show this ability to grow very rapidly in a macrophage model. But the real question then becomes, what are the effectors that are being released that are essential for intracellular replication? And during a comparative genomic paper that we had published, we proposed, like the Legionella system, that there are a number of ORFs that coded for eukaryotic domain-containing proteins, and so these become and have been all tested for their ability to secrete it. And one of the ways that this test is done is through making gene fusions with adenylate cyclase from Bordetella pertussis, infecting a cell, and then looking for the secretion out of a bacterial-containing vacuole. This is a Legionella-containing vacuole. To complement in trans this homogulin activity, and you can see this CYA assay to determine whether it's secreted or not. And so in that system, Craig Roy, as well as our group with Bob Heinz's group, have tested one category of eukaryotic domain-containing proteins pretty effectively for their ability to be released, and those are called anchor and repeat containing proteins. And this just shows the analysis of a small set in Craig Roy's lab, and then within the different isolate groups that we look at, that we had genome information for, the ability of these various anchor repeat containing domain proteins to be secreted. And there was a whole analysis of their plasticity across these different bacterial populations. And one of the important observations that you can make subsequent to the expression and characterization, whether secreted, is where these effector molecules translocate, localize when they're ectopically expressed by eukaryotic cells. And so we then use that as a justification to go through a broad screen to look for a variety of type 4 secretion-dependent effectors. And we used a variety of screening tools, a 2-hybrid system, and some powerful bioinformatic predictions to look for various domains that were associated with other secretion systems. And then we evaluated them initially in Legionella pneumophila, but we've since identified how we can test that in Coxiella Burnetti using the SEAM reporter system, and then functionally characterize them with various assays, including ectopic expression, heterologous expression in a yeast system to look for trafficking defects, and a number of other assays. And so I'll just finish with a few of these assays to show you where this field will, we hope, go. This shows the way that we screen a large collection. This was 32 different secretion substrates that we characterized in the PNS paper in 2010, and this shows the secretion in a wild type, and the power that they have that we had in Legionella pneumophila is we had a secretion defect mutant to make sure that the secretion was type 4 dependent. We then converted, as I mentioned, this assay and a subsequent fusion assay into C. Burnetti because we developed a stable plasmid that could take a gene fusion back and then report in this ratiometric release assay. And subsequent to this, we have created, as I mentioned, secretion minus mutant, and then can compare the requirement of the secretion system for the release in this assay. And this is the first table of substrates that we've identified. And I'll point out, relevant to the topic that I mentioned broadly, are there genes that are responsible for the pathotype variations? And so about 40% of these genes, in a comparative sense, are polymorphic or plastic in terms of isolate groups. And so each of those that aren't fully coding in one of the chronic isolate types makes the candidate to test whether it has an impact on acute disease. As I mentioned, another screening tool that we used bioinformatically was to look for the promoter that is shared by a large collection of secretion substrates as well as the components that code for type 4 secretion element, which is a PMRA. So this is a two-component regulated process, and we were able to identify consensus region based on our screening that allows us to increase the number of substrates that we've tested for the ability to be secreted. So we now have 90 different genes, proteins, that can be secreted that are type 4 secretion dependent in C. burnetti. We haven't completed the entire search, and I imagine that they'll be in the neighborhood of 120 to 140 based on our screening tools. There are actually over 200. There's about 240 in Legionella amorpha. Now, of those 240 in Legionella amorpha, there are probably 20 that actually have phenotypic variations in a particular pathogen model, whether it's in protozoa or eukaryotic cells. And so one of the tools I mentioned that we have been using to focus in on those that might be important is where they localize when they're ectopically expressed. A second is whether they're toxic when expressed in a yeast system and conditionally toxic based on the trafficking of that yeast system. And so we focused on a couple of these. I'll also mention, I'll show you data today, that we have started to accumulate individual transposon mutants in a number of these secretion substrates that will allow us to test their relevance in terms of the requirement for intracellular replication and in terms of causing acute disease in an animal model. And I'll finish with this set of data. This looks at the subset of substrates that localize over ectopic expression to the nucleus. And so those are particularly interesting to us. And I'll point to a particular specific paper that was a pretty interesting study that was done by Ed Shaw and his group, published last year. And what it showed was that C. burnetti infection can lead to an active modulation of post-transcription. And that makes, it's a simple thing, but it was a pretty well done experiment. And that tracked back to a number of inflammatory cytokines that were actively modulated in that experimental system. There were some issues about the way that study was done, but there was also, I think, a lot of positive that you can deduce from that. So I think, in a general sense, we can take from that that C. burnetti does actively modulate the transcriptional pattern that's in response to infection, and good candidates for that modulation would be effector molecules that traffic to the nucleus. And so we think that a couple of these are in our forefront. There are two, and I show you one here. There's a second one that we think that really do have the potential to modulate nuclear responses and transcriptional responses. This one, 1314, actually has a nuclear localization signal associated with it. If you remove that nuclear localization signal, it no longer traffics to the nucleus. And a way that you can look for particular proteins that are able to interact with eukaryotic chromosome is to do a precipitation and co-precipitation experiment called chip analysis. And so we've looked at all 8 of these nuclear localized substrates, and at least 2 of them strongly chip down DNA. And we've begun to characterize the individual DNA fragments that chip this down. And this looks at the readout of 100 different clones from this chip DNA. And you can see the distribution of open reading frames that are associated with the chip fragment as an indication for genes that might be regulated during the expression, ectopic expression of this particular gene. And so with that, I'll just finish by going back as an overview of what I was trying to tell you to begin with. And that is that Coxiella Burnetti is an intracellular pathogen. It infects principally monocyte macrophage populations. It's taken in, eventually delivered to a vacuole that can develop into a large replicated vacuole if there are not macrophage activation signals provided to that. They develop in a lifestyle, small cell, large cell, life cycle kind of growth phenotype. And within that, we believe that there are at least two important secretion systems to release effector molecules that can interact with that vacuole as well as the cytoplasm. And we think that the type 2 and the type 4 secretion effectors that are released will have active modulation of the host response, but upon interferon gamma or another inflammatory signal, those vacuoles become small, non-replicating vacuoles. We believe that they deliver nitric oxide and oxygen rather than that compartment, and that's principal mechanisms of killing. I think I provided all the answers to your questions to make sure that you get them right. And with that, I'd like to thank the people in my laboratory that are currently there, including Chen Chen, who did all of this initial screening for type 4 secretion molecules. I'll first mention my longtime collaborator and happened to be spouse, Laura Hendricks, as well as Mary Weber, who's taken on this type 4 secretion system with vigor. And I also want to note really important collaborator. Xiaocheng Liu is a type 4 secretion guy working in the Legionella and Haemophilus system at Purdue University. Bob Heinz and I have had the pleasure, as well as Paul Baer, working with for the last 25 years. And I want to end and thank all the inviters for my
just a few words, because I'm going to run after this to go to Paris. I just want to say that at the moment we were the only lab, this of Jim Samuel and my lab in the world working on fever. Nobody was working in the US anymore. We were probably the only place working on this in Europe, and for a long time he was the only one working on overseas on this. And one of the questions that have been really fascinating this last time is the strain-specific clinical manifestation. I think that, of course, there is something with the strain that is related. But I think that it's come since the very beginning. I think that there is a confusion, and the confusion is reaching because of the Netherlands outbreak, which is to discriminate between acute and chronic. I think it's not right. I think that acute infection, I believe I understand approximately what it is, even if I don't know why some get pneumonia and some get hepatitis. But there is no such thing such as chronic infection. There is endocarditis, that's something. There is chronic vascular infection, which is something close to endocarditis. And there is some sporadic things that we don't really understand. And I think abortion is one more other study. And trying to group these all together, make a concept that become, when used from people not really familiar with QPV, become incredibly confusing. And this has been part of all the Netherlands start, where they really try to define chronic Q fever without referring to a specific clinical involvements. That does not make sense at all.
So it does, and I appreciate it, and I'm happy for you to make that clarification at all times, because I never in graduate school, because you can say things in graduate school that get misinterpreted, and I never tried to associate a particular chronic disease versus. I just pointed out the differences phylogenetically, and I think there's a lot of difference in terms of virulence potential between the two.
Yeah.
And the development of chronic disease is extremely complicated, obviously. And as you're pointing out, we don't have the opportunity in the U.S. to look at these cases because we don't have the population base. We have essentially none. There have been 5 cases that we can actually get clinical reports that came from the United States, and then 3 cases from Nova Scotia, which we had access to. There are no other cases. And we think it's all because of surveillance. So again, the center that you have here is critically important for identifying these patients. We just don't have the tools in the United States to identify them.
And one of the very last things that is really very interesting that we just have been involved with, and maybe you have been one of the reviewers, is that we've been really surprised. Here we've close to Marseille, there is a place where the reason why they find the first case of abortion with Coxiella Burnetti. It was a nurse. She was treated by the chief of the internal medicine, and the nurse was working in the hospital, and the obstetrician was infected. So all the hospital became mad about abortion and fever, and then they are actively looking for this. It's Martigues, it's Montagne from here, and now they get plenty of cases just by seeking and seeking. So we have quite, this is probably the only place on the world where there is an active surveillance and fever, acute fever in pregnancy. So we arrive at such a level, and we determine a strategy to treat them. And when comes the outbreak in Netherlands, in fact, they never get any case of abortion with acute fever. But it's global. And then we go back and analyze all the strains that we have that are being linked to abortion. And of course, there is a strain specificity, and specifically the one that is more associated with abortion of all is that with the plasmid QPVD. Okay. And this one is highly significantly linked to abortion in human beings compared to all others. So I believe that the strain is not abortive. Which is, I think, the first time that we get an outbreak of Q fever with no abortion, but because this clone, this bug was not aborted. So this is one more thing. So it's really coming out that there is a strain specificity, of course.
I didn't review the paper, but I would like to see the paper. Other questions? Yes?
Can you describe the best evidence of the different strains by aerosol?
By oral?
Aerosol,
Aerosol. Did we look at this? Yes, we did, in fact. The guinea pig is an aerosol route challenge. They're delivered in an aerosol using a Madison chamber, which creates droplet nuclei. Although this isn't a disease that's transmitted person to person, I think aerosols are actually dry aerosol. But at least you get down to the deep alveolar tissues in that infection model. And so in the guinea pig, we did it by aerosol infection. And that's where we saw a significant difference in pathology. And in the mouse model, we've also done using the Madison chamber aerosolized to the lower lungs. And when you do it's not so pronounced when you do it by intratracheal, because intratracheal instillation actually drops a big drop of liquid into that space, and it's a pretty good model, but it's not the best model. But yes, we do see that difference in disease process in the mouse model, the guinea pig model, by aerosol. Thank you. Apologize for not speaking French.